Okay, hi. So today we're going to go through A3.1 and A3.2. So this is our unity and diversity of organisms. A bit of a strange placement because it's in the organism section with all the body system stuff. But this is not really looking at body systems. It's really just kind of classification. Cladistics taxonomy would be kind of the best explanation of what this little section is. So we'll start with A3.1. A3.1 is SL and HL, whereas A3.2 is HL only. And so we're going to kind of, the first section be everybody, and then there'll be a little HL section at the end. You don't need to write super tiny. These topics are not quite as full. So feel free to kind of write at your slightly more normal size. So this section, unity and diversity, and really it says unity and diversity. Most of this stuff is really about diversity. So really looking at what are the difference between different types of organisms. So we're going to do a little bit of taxonomy here in terms of how are organisms grouped. There's more of that for sure for HL. So we'll come back to that in the second part of this. But for SL, you still kind of want to have some idea of kind of the grouping of organisms. But then we're really going to zoom in on species and really look at kind of what that means. So it's a little bit all over the place. So we have basically this diverse group of organisms. And so we have all these different organisms, and then we need a system to divide them. So this has been around since the 1700s, this idea that we need to in some way group things because of the sheer amount of kind of complexity there is. So there needs to be some way of organizing the intense amount of diversity. And so they are grouped, diverse groups of organisms sorted into. And so you do need to know it's a distinct dot point for HL. For SL, it's not quite as distinct, but I would still say it's worth knowing the categories. So if we kind of categorize it from inclusive, so when we're saying inclusive, it includes the largest group of organisms. And we will include domain, because HL, you definitely need to know domain. So it's going to go domain to kingdom. Kingdoms are then going to be divided into phylum. Phylums are going to be divided into class. Class is going to be divided into order. Order into family. And then families into genus. And genus into species. And species then will be your most exclusive. So this is going to be your smallest grouping that is exclusive to only a fairly related, similar group of organisms, and we'll talk about the definitions of that in just a second. Definitely worth coming up with some mnemonics. So I say, did King Phil come over for great soup? It's not the world's greatest one. You work out what works for you, but it is helpful to have some memory clue that helps you remember the ordering of them. Most of you are going to be fine with genus, species, kingdom, phylum. It's these ones in the middle that get a bit confusing without that. Okay, so like I said, those are the groups. You really don't have a lot you need to know. In the old course, there's a little bit more, like you had to actually know the names of the phyla for plants and so forth, but that's not really in it anymore, so that's good. So you do need to know a little bit about how this is used. So genus and species, that is what is used for binomial nomenclature. Which is essentially the assigning of scientific names to organisms so that we have a universal way of referring to them that's not changing based on every language to ensure that we're referring to the same organisms. So binomial nomenclature is just scientific naming. It's usually Greek or Latin. So it has some sort of kind of origin, historical origin, and that's what's used. Just remember, if you're using it, you're doing it italicized. That's my attempt at italicizing. And that your genus should be capital and your species lowercase. So just make sure if you were ever writing that like in a data question that you know how to write that. Okay, so from there, like I said, that's kind of the brief intro. Then this topic really looks more at the idea of species. So we'll go into a little bit about, okay, we have all of these different organizations. How do we arrive at the idea of these are a species? And for my class, we've done this kind of out of order because we already did speciation in A4. So because we haven't gone linear, it's probably like a strange placement, but that's all right. It's good to revise all that anyway. So when Linnaeus made this system, he actually just said species are the most morphologically similar. So the kind of previous, or the way it was originally done when it was done in this session here, is just based on the most morphologically similar. So these are the ones essentially that just look the most alike. As you can imagine, though, that becomes really subjective as to how similar is similar enough. When should we divide it? All you have to do is look at, we've used the example before, but look at dogs. So many different breeds, that's partly because of our doing, but nonetheless, huge morphological differences, same species. So where do we draw that kind of line? So that is kind of the old way of doing it, but not very accurate because of those kind of subjectivity issues. So now what we do instead, and again, we have already introduced this when we talked about speciation, but just a reminder that we call this the biological concept of species. That's just the official term for it. And the biological concept of species is all related to mating. So if we recall, if you are a shared species, they are able to successfully reproduce. And what do we mean by successfully? They need to be able to reproduce to create fertile offspring. And the fertile is really important because otherwise there are some different species that can produce sterile hybrids. So for it to really be one species, they have to be fertile. So let's talk a little bit about what the benefits but also limitations of this as a concept. So we can look right away and say, oh, that's good because it's not as subjective, so we can actually have like a test for it. So rather than just be like similar enough or not, we can say able to successfully reproduce or not. The other benefit of this is that it actually is a reflection of what we now understand is genetic similarity. So it's not directly saying this amount of DNA similarity, but the ability to reproduce and create successful offspring stems from genetic similarity. So it is a mechanism, an objective way of kind of creating a cutoff for kind of being genetically similar enough. So that's kind of successful reproduction is the result of genetic similarity. And even more than that, it's the result of something even more specific. So when we say genetic similarity, in particular, it's related to kind of chromosomes. So chromosomes need to be aligned in number, but also aligned in kind of similarity so that they can form homologous pairs. So you would need to have the same number, but also enough chromosomal similarity that when the two gametes come together, the chromosomes can form homologous chromosomes. That doesn't work if you have different numbers. So an example of how this is a reflection of that is that in order to successfully reproduce, successful reproduction requires essentially this exact same chromosome number. So it requires the same chromosome number, as well as kind of general chromosome similarity. So we're going to focus on number, but we do want to recognize it's more than that. If you had two that just happen to have the same number, but their chromosomes are completely different banding patterns and so forth, they wouldn't form homologous pairs either. Okay, so that's kind of a core similarity. So we're going to use that in a minute as our springboard to talk a little bit about karyotypes. So how do we actually compare chromosome numbers, and how would we use that kind of tool to assess whether things have similar chromosome numbers or not. Before we do that, I want to do a little HL bit, so SLs just chill for a second, or fast forward till you see me writing about chromosome number again. But I want to look at, are there still negative things related to this concept? So we can say, oh yeah, it's definitely better, it's now much more objective, but what are the things that are still problems with the biological species? So we're going to look at kind of 3 challenges for this definition. And then we are going to make a note that this is HL only. Okay, so the HLs you need to essentially be able to identify as concerns with this is that one thing is it works really well for sexually reproducing organisms because the test is, put them together, is their offspring successful? If you have asexually reproducing organisms, each individual makes their own offspring. So does that make each individual their own species? Technically. So a lot of times then they almost have to go backwards to morphology and they have to actually look at that as kind of the determining instead. So problem number 1 is asexual sexual reproduction. So if we used that definition, we would end up with just way too many species. So the result would be too many species. So then you end up going back to kind of grouping them based on similarities. So that's a group that this definition doesn't serve well. The other thing is that, again, the definition assumes what we call vertical gene transfer. So offspring or parents pass genes on to offspring. So if the parents are able to pass on genetic material successfully to the next generation, that's this definition of species. What it doesn't account for is when you have gene transfer within a generation. So you have one adult crossing and giving DNA to another adult. Well, then if they're able to do that successfully, does that make them a species? Or it makes it difficult to find species because suddenly they have new DNA introduced not through reproduction. So the term for that is horizontal gene transfer. And that is another thing that can make this concept difficult, is because it can end up kind of causing genetic similarity between organisms, but it's not due to reproduction. And so just to remind you, that is adults transferring genes. And it's really common in bacteria. So this is a real concern when trying to identify species of bacteria. Both of these, because a lot of them do reproduce asexually as well, and then they transfer this, so then where do we say this is a strain and this is a different species? So it gets really complicated there. And then the third one, which is true of all things, is that we have talked about speciation, and there are some sudden speciation events, like things that are the result of polyploidy, but your average speciation is generally due to the introduction of a reproductive barrier and then some sort of gradual loss of reproduction, and therefore they diverge over some period of time. And so speciation is generally gradual. And so at what point is it called, essentially? So at what point is it they rarely reproduce together? Are we starting to say they're separate species? Where do we kind of draw the line that says reproduction is getting further and further apart and they're diverging and gene pools are becoming separate? So that's the kind of other factor, is that this is binary, whereas the process of creating new species is not. Okay, so that is a little bit of the concerns. That's really all you need to know. So we're going to go from that now back into this idea of chromosome number. I'm just going to delete. So we have the board again, but we're going to look at how do we determine these chromosome numbers. Okay, so we're going to look a little bit again at how do we compare these things. So we're going to look at how do we compare chromosomes, and then how do we compare genomes. So if we have compared chromosomes, then we want to go into a little bit more in depth and say, okay, yeah, we know chromosome numbers are similar. How are their actual genomes different? Then that requires a different technique. So we'll look at both of these. So first thing we're going to do is we're going to look at how would we go about comparing chromosome number. All right. And this is this funny one that the IB has this kind of very specific wording for. So they really like this term karyogram, and that is in some way distinct from a karyotype. It's not a common distinction. Most sources are just going to use karyotype. So I'm going to use karyogram because it's in your course, but if you're ever reading elsewhere, you'll just generally see the term karyotype. But essentially the idea is that what you want, if you want to make a comparison, is you want a karyotype. So a karyotype is basically information on the number of chromosomes. So this is basically information on the number and nature of the chromosomes. So if you were to know someone's karyotype, you would know how many chromosomes they have, if they have any chromosomal abnormalities, what their sex chromosomes look like. So it's information about their chromosomes. And that is actually created by a karyogram. But again, a lot of times the karyogram is just called a karyotype. But that's the distinction. So this is the tool that is actually used to make the karyotype. So what a karyogram does is it produces a photo of chromosomes inside cells. And the cells are going to be cultured, so they're going to be reproduced and then also given treatment to initiate cell cycle and then arrest it. So it's going to be of cultured cells that are in mitosis. So we want to be able to see the chromosomes. Remember, if it's not in mitosis, chromosomes are not visible enough to be seen. And then you're going to actually gain the photo by looking at under the microscope. So those are kind of your key points when we look at what is a karyogram. It's a photo, it has to be visible chromosomes, therefore it has to be in mitosis, and it's generated from looking at them under the microscope. So then once you have all these chromosomes, because in mitosis, homologous chromosomes do not come together, they only do in meiosis, but we would not want to use meiosis because it'd be much harder to access gametes, and also because they will start crossing over and then that confuses things. So we're going to use mitosis, but they're not going to be in homologous pairs, so we're going to have to put them into homologous pairs. So they're going to be sorted into homologous pairs, so that we can kind of double check the number. So we want to make sure we're going to pair them up, and then we're going to essentially check the number of them. So the way that they are sorted into pairs is that is going to be based on the size. So the pairs are going to have the same size. They're going to have the same or similar, they should have the same, banding pattern. And they should have the same centromere position. And so then when they are presented in a karyogram, the karyogram that tells you the karyotype, they're generally organized from largest to smallest. So what you might see is these two were obviously homologous pairs because they're the same size, so that might be pair 1, and then that's going to go all the way down to what might look more like that, and that might be like pair 23, something like that, 22. X would look different. So they're going to be organized in kind of size order. So they're going to be grouped by their homologous pairs and then organized into their size order. And then you can look and say exactly how many pairs do they have and work out their diploid number. Okay, so then we're going to look at kind of one application of this. So a finding that was done by using karyograms to better understand the relationship between organisms. And that is looking at the specific example of human chromosome 2. So this is an example of a karyogram finding. So this is a named example, so it is in your syllabus that you could have to present the evidence for this, so that is why we are going to make sure to go through this specific one. So basically what was done is they actually did karyograms to look at the difference between humans and chimpanzees, because they're believed to be really close ancestors, but we don't have the same chromosome number. So the question was, where did the chromosome change? Chimpanzees actually have more chromosomes than humans, so particularly how do we kind of have an evolution towards complexity but a loss of chromosome? So the hypothesis is that human chromosome 2 is actually a merging of two different chimpanzee chromosomes, so 12 and 13. So we're going to draw that just to visualize it, and then you do need to be able to identify 3 pieces of evidence for it. So let's look, if we looked at the example of the chimpanzee ones. So let's stay there. Let's give it some bands. I am just drawing arbitrary bands. I am not an expert on the banding pattern of chimps, just to be clear. So that's going to be chimp chromosome 12. And then this is going to be like this. Chimp chromosome 13. But we need to add a few extra things that are going to be relevant. So first of all, I probably should have drawn it in a different color to begin with, but the actual centromere has distinct DNA. So it's called satellite DNA, and it's non-coding DNA. So it's often just repeats, short repeats over and over and over that do not ever code for amino acids. And it is quite distinct. So you can usually pick out centromere DNA. And then the other thing they're going to have is some telomere DNA. So at the tops and bottoms of the chromosomes, you're going to have these specific kind of, repeat satellite sequences that are associated with telomeres. So those are both going to be important when we look at the evidence. So what appears to have happened is that these two then kind of merged, because if you look at human chromosome 2, so I have to be careful, try to draw it accurately. Let's do it like that. It's going to have these same banding patterns up in the first arm that are similar to the chimpanzee chromosome 12. And then if you look at the banding pattern then here, the bottom half bears similarity, like that, to chromosome 13. So it ends up looking like the top banding patterns match chromosome 12, the bottom arm banding pattern matches chromosome 13. Additionally, it retains this centromere, but then there's some random centromere DNA in the actual arm. So that's centromere DNA in the arm. And what that suggests is that this centromere just got merged into it. Similarly, there is of course telomere DNA at the top and the bottom, but on top of that, there's this extra bit of telomere. I'm trying to figure out where it happened. It's like right under this one and a bit like under this one. So there's some extra telomere DNA in the arm as well. And you would not normally see those things. So those things suggest that we are looking at some sort of merging. And then if you were writing the evidence, don't forget to just also note that the banding patterns are very similar, the same. So those would be kind of your 3 bits of evidence if you were providing it, is the banding patterns in the top and the bottom matching those different chromosomes, and then the fact that you also see those kind of satellite DNA bits out of place. Okay, I'm gonna finish try to figure out if I can do this. I think I can. I'm gonna do just the little bit of last bit of HL or SL stuff, and then we'll move on. So this is all looking at comparing chromosome number. If you wanted to do a more sophisticated analysis, you could actually now compare the entire genome, and that would give you more information, because just the number is not going to be quite sensitive, but like enough for what we want to do with cladograms and things like that. So this would be comparing chromosome number. But the other tool we can do now to look at different species is to compare genomes. Okay, so we need a different tool for this, and so this is a more complex tool, although it has gotten much, much easier. So what used to be very challenging to do is now much easier. So that is whole genome sequencing. So essentially being able to derive the entire sequence of genes, of bases, or A, C, T, G, A, C, T, G, 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 C, A, all of that from a single organism as a representation of their species, or you can compare organisms within a species. And so that used to be really difficult, but post the Human Genome Project, so we do want to acknowledge that the Human Genome Project played a big role in this. this process is now much cheaper and much easier and quicker. So what would have been much more challenging to do in the past is now quite feasible. So we're going to be able to do that. So essentially it wants you to look at kind of current use, which links to what we've been talking about. But then we also want to quickly note that there is kind of a potential future use, which is less related. So in terms of current use, the current thing that this is being used for is for what we've been talking about, is to look for evolutionary relationships. And we'll kind of clarify that more in just a second, and then it will also become really important for HL. We get a little bit more into cladistics in the second part of this video. So we can use genome sequences to really work out exactly how different they are, because again, chromosome number is not as sensitive, and some different species can end up still having the same chromosome number. So this would give us a better insight into their genetic similarity. So this is kind of an indirect one way of looking at it. This is much more specific. One potential future use that is future use, but it is already happening, but is still kind of being developed, is to use this on an individual level for real personalized medicine. So the idea here would be that essentially you can use it for diagnostics, but also even for like as pharmacogenetics. So essentially knowing what medication to give someone because you actually know their genetic sequences. So using that to provide like tailored medication. So that's just listed in your syllabus. I don't think you necessarily need to know details about it because it's still in the work. But essentially the two main things it would be used for is diagnostics and treatment. And being able to do kind of quick, efficient diagnosis, and then being able to do tailored treatment that we know will respond to your specific mutation and things like that. So that's kind of an exciting place where it's going. If we are looking at it for these evolutionary relationships, the main thing that you just need to be able to account for is if you do this, what is the difference kind of within a species versus between species? So essentially, what is the key finding of this? And the key finding is that if it is within one species, so within one species, you should find minimal differences. So they should basically be almost the same on a genome-wide level. So if we look at individual kind of phenotypic alleles that have polymorphisms, we might find more. But if we look on a whole genome within a species, they should be highly, highly similar. So we should see minimal variation. And we should see much less than 1%. So I think it's somewhere about 99.9 in humans. So a fraction of a percent if we look across the whole genome. And that 1% is due to single nucleotide polymorphisms. You can write that out if you don't know what that term is yet. But basically that means that one single base is the only difference in the whole gene, but it leads to allelic and phenotypic differences. So you could have a gene that's like 500 base pairs long, and yes, you end up with variation at a phenotypic level, but it's actually only due to a single nucleotide being different. That's how you end up with this less than 1%, because even where there are variations, it's due to such small changes at a DNA level. Okay, so that's what you would find within a species. If you were to compare between species, which is really what we're talking about, you are going to find two things. First of all, you're going to find way more variation. So even organisms that are closely related, you should find way more than 1%. So you're going to find 2, 3, even between closely linked ones, all the way to 50% and onwards. So first of all, we're going to see more variation. But also the amount of variation depends on the relatedness. So it's going to depend on kind of how similar of an ancestor do they have, how recently did they separate and speciate, and so forth. So those are the two things that we would want to know, is that we would expect much more. So those hopefully are pretty straightforward understandings, but that would be just how you could use that. All right, the last little bit is HL only, and in fact, A 3.2 is all HL, so SLs you are done. Goodbye. Enjoy. HLs we need to do a bit more. So 2 more things from this kind of subtopic, and then we'll go from that and move into A 3.2. So I'm just going to erase. All right, and these don't have any amazing kind of similarity between them, so I'm just going to label this bottom section HL only. And just in terms of context, this doesn't matter because it's however much you have, but that's how much I use, and that was being generous, so just use whatever space you have left. Okay, so the 2 different things that come under this HL banner for this topic is dichotomous keys and then eDNA. So let's do dichotomous keys first and talk a little bit about those, and then we'll wrap up with the eDNA. Okay, so what is the purpose of a dichotomous key? So a dichotomous key is going to be used to identify and usually name. So generally, if they're good, they're providing you the scientific name. So when we say identify, it means not just like common name, but hopefully, if it's known, it will provide that. So it's to identify and name an unknown organism. And this is where it's a bit tricky because we're going to look at eDNA, which actually does it by its DNA. So when we use a dichotomous key, we are doing it based on morphology. So even though we've kind of said morphology is bad, it's not objective enough, dichotomous keys are using it based on that. So it's identify and name an organism based on morphology. And the dichotomous comes from the idea that it's using steps with 2 options. So it uses steps, each with 2 options. So it does say in the syllabus that you could have to create one. So you're supposed to create one by engaging with local flora and fauna. What that means on an exam is they're going to give you some pictures and then you would have to make one. So you could have to design one or you could just have to use one. I'm just going to give a brief example. If you feel like I'm good on this, you don't have to write this part. But again, just because it is listed that you could have to do one, I just thought we'll do a tiny little example of just how to format it. And I'm not going to use scientific names. I'm just going to use common names, but just to make the idea. So you would always have numbered steps. So this would be like 1, and it's going to have 2 statements. So I'm going to say like has feathers, and then lacks feathers. But what you'll see is I cannot say if it's a true dichotomous key, has feathers, has fur, has scales, has slimy skin. It kind of seems dumb because it feels like that might be a faster way to go about it, but a true dichotomous key only has 2 options at each place. So if it has feathers, then it would tell you, oh, good job, you've identified it, it is a bird. If it does not have feathers, it's going to tell you to go to step number 2. Then you'll come to number 2, and now it's going to say has scales, lacks scales. So if it has scales, it's going to now say, good job, it is a reptile. If not, it's going to say go to 3. And then when you get down to 3, it's going to say has fur. And now if we only had 4, you could say lacks fur or has slimy skin. This is a very fancy one. And then we would say mammal, amphibian. So that's a very basic example, but the point is to only have 2 options at each place. It could be 2 different numbers, so it doesn't necessarily have to say reptiles. If there were multiple reptiles, this might have said go to 4, and then it would be things like has legs, does not have legs, and then it would be snake, crocodile. So it doesn't have to have a named one at each step, but it can. Okay, and then just remember, if you're using it, you need to always start at step 1. And of course, this is a very, very simple example, but in other examples, if you don't start at step 1, you'll get confused because they may sometimes repeat a step in 2 different pathways. And if you just kind of go straight down, you'll end up with the incorrect identification. Okay, so that's if you had morphology, so that's pretty easy. Like we could in theory make one and use it pretty efficiently. But again, it's based on being able to identify features, which with organisms that are really, really similar can get more complicated. So another way of identifying organisms is instead to use eDNA. So we're gonna look at eDNA and barcode libraries. Okay, so let's start with kind of defining eDNA. So eDNA stands for environmental DNA. And so environmental DNA is DNA that is gathered from a habitat. So it's DNA gathered from some sort of habitat. So a lot of times it will be soil or water. Those are probably the 2 most common ones, but you could come up with other habitats where that might be sand, things like that as well. And then the idea is, where did the DNA come from? And so the DNA would be a collection of things like hair, dead skin cells, debris, kind of like dead particles. It could be waste. So it's not whole organisms usually. It could be with microorganisms, so you could actually gather an entire organism, but a lot of times it's shedded DNA that's just found in the environment. So then let's have a little look. So let's say here's my spring. Woo, that's very dramatic water. And so if you gather an eDNA sample, generally speaking, there's going to be multiple DNAs inside of it. And again, you're not seeing the whole organism. So you're just getting kind of fragments of DNA inside of this. So the general thing that this is used for then, so like why gather it? So the most common use and the use that we're going to talk about is to actually use it to assess the diversity of the ecosystem. So one of the ways we talked about biodiversity in A4 is you can look at it in different ways. And in particular, what we're generally looking at is kind of at a species level. So we're looking at kind of species biodiversity, not ecosystem like not different ecosystems, but that ecosystem's species biodiversity. So that's generally the goal of what we're looking at, is how many different species there are. So what we're going to do is we're going to collect the DNA and amplify it with PCR. Because we're not going to have a lot of it. So we're going to make lots of copies with PCR. And then there's essentially 2 different ways of going about doing this. So one option is that you can then identify all of the species. So we would be going through all, I only drew 3 there, but there might be 20 in the sample. And we're going to go through and identify all of them. And in order to do that, you're going to have to use DNA barcodes. So we would basically be running each one through a DNA library. So that's going to be stored in a library. So it would be like if I took you to a supermarket and I gave you boxes of things that were wrapped, I don't know why, but the barcode was still free. And so I was like, identify this, and you would scan it and then it would come up and it would say Oreos. And you'd be like, got it, I identified what it was. So that would be the idea, is that you scan an unknown thing, and then when you scan it, it shows up and tells you what it is. So remember, the DNA barcode is based off of a specific DNA fragment to that species only. So that DNA is specific to that actual species. So when you run their DNA through, it'll find that barcode in it and be like, da-ding, and tell you that that's what it is. So it's a little bit more tedious, but it would give you an actual sense of exactly how many different species there are in the area. Just two limitations worth noting is that it will only tell you present, but not the population size. So it's good if you're just doing a count of the actual ones, but it's not good if you're trying to actually work out like if they're one dominant species or not. And the other thing is that DNA can break down, and so you could be missing things. It's not a guaranteed that you're catching all of it. But when we say all species, one benefit is that some of the DNA could be from microorganisms. And generally, those are a lot harder to do if we're doing morphology visual checks of an ecosystem, if we're counting species that way. So that's a real benefit. The other option, we'll just do quickly, is that instead of looking for all the species, we can get an indication of biodiversity just by looking for indicator species. And so instead of trying to work out exactly how many there are, we can look for species that we know. It's kind of a little bit different from biodiversity, but basically if that species is present, it indicates that the ecosystem is healthy. So if present, the ecosystem is healthy. So that's kind of another tool that eDNA can be used for. It's a little bit faster-ish, although you're still having to basically scan and look at if any of the DNA is in fact indicator species. So they kind of just tell you different information. This is going to tell you how many different species there are. This is going to tell you kind of if pollution levels and so forth are removing some species from the area. All right, so that's A3.1. HL, we're going to go on and do A3.2 as well. Okay, so we're going to pick up with HL. So again, this whole thing is HL only. So turn away if you are SL, unless you just love cladistics, in which case feel free to join us. So this is continuing on with classification. So it's really just kind of a continuation of some stuff that we drew up there. So in the last one, we kind of talked a little bit about this idea that we group diverse organisms into these things. I do want to kind of pick up another point here, which is kind of why. So why is classification so important? So if we're talking about classifying, which is to essentially group organisms. So classification kind of includes the grouping and naming of organisms. So the question is kind of why do we do that? What is the benefit? And essentially the short answer is basically communication and organization. So what we want to recognize is there is an immense amount of diversity, and if we don't have any system to organize and sort them, it's basically like why you would put, documents in folders on a computer. Otherwise it gets really difficult to access and share information. So essentially we just have immense diversity, that's the IB word. So we would just have bazillions, don't quote me on that number, millions of files, and if we don't have any way of organizing them, then if we're like, oh, I found out a new bit of information, I want to add it to this Word document, and there's just stuff everywhere. Whereas if we have it folders within folders, it's a lot easier to find and add information. So essentially what classification does is it facilitates two things. So it facilitates first organization. So it has a reason for where we can kind of access information. And then if you have an organization system where you kind of know where different things sit, then that allows for communication. So this way we say, okay, I now know that we are all talking about the same fern because we're using this name for it and we know it belongs in this group. And if we do that, then that allows for kind of cooperation and broader study. So it makes it easier for scientists to communicate about organisms, and then that enables them to collaborate and work together and so forth. Okay, so then the question becomes, how do we classify? How do we group organisms? And there's essentially the old way, and then we're going to look at kind of the new way, which is already what we had started to talk about last part. We're going to kind of expand on it. So the old way from the 1700s was this idea of domain, kingdom, phylum, class, order, family, genus, species. So that is called fixed taxa. So what that means is that every organism has to belong to each one of those, and they have to go big to small. So that's what we mean by kind of fixed. So for every organism, there has to be a domain, and then you have to go all the way from domain down to species, and they have to go in order. So genera have to be bigger than species and so forth. So have to belong to all, always have to go in the same order. So we're gonna kind of cover what is not so good about that. And it's related to what we kind of talked about with species, but we're focusing more on morphology. But the other idea here is that when we just group them, it became a bit arbitrary. So like, when does it become a separate family? So at least with species, we have a reason, like reproduce or not. But when it was like, when do these two genera become so distinct that they're no longer the same? So it becomes really arbitrary. There's not any sort of meaningful way of determining the other taxa. So kingdom's a little bit better. So kingdom, we can kind of do it based on like cell wall makeup and certain things. Species have a definition, but a lot of the ones in the middle don't have clearly defined characteristics. So it becomes a bit, it doesn't really have any meaning. It just becomes a bit, they decided this now looks different enough to separate them. So that's the first problem. And then again, it kind of comes back to the idea of what we said about speciation, but it doesn't represent the idea of gradual variation. So these three organisms have variation between them that's gradual. When do we draw the line and say different class? So it doesn't reflect the idea that variation is gradual. So it kind of makes it so that it seems like it's really cut and dry when they're different enough and when they're not. So those are some of the kind of concerns with that system. So what we need to do is essentially develop a whole new system. So it wants you to kind of associate this word with it, which is paradigm shift. So what we're saying is we needed a whole new way of thinking about it, not just like tweaking it. So you can tweak it, like adding domains, changing the biological concept of species. Those are kind of tweaks to an existing system. But the other way is to create a whole new way of looking at grouping organisms. And that is with using instead cladistics. Okay, so what is cladistics? So cladistics is grouping organisms into clades that are unranked. So you could belong to this clade, but you could also belong to this clade, and one isn't bigger, more inclusive necessarily than the other. You can belong to multiple clades, and they're not ordered. They're just like, this is a clade you belong to, this is also a clade you belong to. So grouping of organisms into unranked clades. So that's where cladistic comes from. It's also where cladogram comes from. So let's define what we mean by clades. So what is our definition of a clade? So a clade is a group of organisms that share a common ancestor, so they can link to having a common ancestor. And this is why we say it's unranked, because we could argue all life form has a common ancestry. If you go to Luca, last universal common ancestor, A4. So again, technically everything could be a clade, but it just essentially means you're looking at a group that you can definitively track to having a common ancestor. So it is just a group of organisms that includes their common ancestor. So as I said, you could say all or life form has common ancestry, but if I said like humans and crocodiles, well, we're not a clade because there's no inclusion of something that is a common ancestor. But if I listed every animal, so human, blah, 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 fish, amphibians, and listed all of them, then I could say that is a clade because those are all animals and they all share a common ancestor, which is like ancestral animal. But I can't say just humans and reptiles because I'm not including everyone with their common ancestor. So it kind of basically means it has to be all inclusive. So that's probably all organisms of that same common ancestor. So it needs to include all descendants of a common ancestor. So the common ancestor could be a primate tree and it could be a clade of primates. It could be an animal tree and a tree of animals, but it needs to include the whole tree, not just random parts of it. So hopefully that makes a little bit of sense. We'll look at it more as we draw some things. And so because of the fact that they do share a common ancestor, they therefore also share morphological similarities. So this is where it gets a bit tricky because you can look at identifying clades based on morphology, but it's an indirect measure. So the best way to do it is based on DNA evidence because then you're actually using the true nature of it. But you can actually look at these kind of morphological similarities and from that kind of group organisms. So these morphological similarities we call derived characteristics or characters. And these tend to be ones that essentially some of them have in common. So you can kind of look at who has more in common, and therefore you can say this group all has a derived character in common, therefore they are in a clade. So that is one way of doing that, but the idea here is that it is due to genetic similarity. So the better way of doing this would be to actually use DNA. So DNA is a more accurate way of identifying them, but you can also do it through morphological. It's just not as objective. Okay, so we'll come back to that in a second because we are going to talk about how you group these. Two things quickly on why is this better. So we had this paradigm shift and they basically address kind of the things here, but the first one is that it is actually meaningful. So it's not arbitrary. So rather than just saying you're in a group because, I don't know, you look more alike, we're actually saying because you evolved from the same common ancestor. So it actually reflects evolutionary patterns. So it actually means something. And then similar to this, kind of related, is that it is actually predictive. So if you know what clade an organism belongs to, you could actually predict their features because of that. So it allows for kind of, provides information on an organism based on its clade. And again, that's because it's not as arbitrary. So you could, in theory, kind of look and say, oh, they're probably going to have some morphological similarities because of their kind of evolution together. So those are some of the benefits of using it, is it essentially just has more of a meaning and a use. Okay, so then how do we actually determine it, which is kind of what we've already talked about. So if this is what it's due to, then the best way of actually determining clades is to actually compare them genetically. So you're going to actually compare them genetically. So you're going to do it by molecular similarity. So you're going to compare them based on their molecular similarity. And of course, the molecular similarity is what causes these. Sorry, there's a lot of lines here. So what we're trying to show is these are all related. So evolutionary relationships cause them to have more morphologically things in common. Evolutionary relationships cause them to have more genetic similarity, therefore more molecular things. So again, you can use morphology because it's an indirect thing, but ideally you're going to do the molecular similarity. This would be the preferable way of doing that. And the way we're going to do that is two different things. And so the first thing you can do is you can actually just compare amino acid sequences. This is often done for less related organisms that have more differences. Historically, it's been a bit cheaper. There's less things to sequence. Generally, it's easier to identify amino acids, so that's kind of the easier one to do. It doesn't necessarily work as well with really closely related organisms because they may have all their amino acids similar, but you could pick up on differences if you start looking for silent mutations. So the other way to do it is to actually compare the DNA base sequences. And when we do that, we are using often a technique called hybridization. And what we're looking for is essentially strand one from one species, the complementary strand from another species, and we're looking for any places where they don't hybridize. So if they cannot stick together, that means they're not compatible, and that suggests there's a difference there. Okay, and so again, if we do this, both of these are good, because they are objective, so they are clear. We can actually say, yes, this is the number, and they also provide quantitative data. So we're basing it, again, on kind of numbers rather than basing it on who has jaw bones and who doesn't, which is how some of them have been done other times in the past. Okay, so then I'm gonna erase because then we're gonna go into kind of what are some uses. So once we know these clades, and this idea, what are some ways that we can use cladistics? Okay. So two kind of uses for it that you need to know about. We're going to spend more time on cladograms, but also molecular clock. So cladograms. And these are often used together, i.e. working out a molecular clock, which is essentially a rate of change. You can add more specific dates and stuff to cladograms, so those are added more to phylogenetic trees. Cladograms tend to be a little bit more focused on smaller groups and looking inwards at that. So again, you could do a cladogram of a very large clade. Most of the time they're done on slightly smaller clades. So you're taking a clade that have a common ancestor and studying all within that common ancestor. Generally, if we're looking at tons and tons of groups, we might use a phylogenetic tree. They just branch a little bit more and they tend to be a bit larger scale, whereas cladograms tend to, again, be very much zoomed in on a specific group and looking at what kind of their differences are. So let's look at what a cladogram would look like. Again, this is one where you could have to interpret or draw it, so it could show up either way. So it's essentially a diagram of a single clade. And remember, what makes it a clade is the shared common ancestor. So that's going to be down here, and that's going to be called the root. And the root has to be the common ancestor of that clade. So this is the common ancestor of all members of this clade. So it depends what group we're focusing on, but if I have a list of 8 organisms, what's kind of the one that evolved initially, that they all kind of evolved off from. So that's going to be down there at the root. Then every time something branches off, those are going to be called nodes. And this is going to be anytime there's kind of a branching off or separation of species. So basically, like, here's the common ancestor, everything's related, and then some sort of speciation event happens. And now we have a new species that is distinct but still shares that same common ancestor. And then that's going to end all the way, so you're going to keep having ones that come off. A lot of times this one here will be called the outgroup, and that just means that of the group, these are less related. So they evolved separately, and that usually means that they're going to have more DNA differences. So these guys are going to have more DNA differences down here, and then as you go up here, they're going to have fewer DNA differences. So the further away down at the bottom it is, the less similar it is to the ones at the top. Then these here are going to be called terminal branches. And the terminal branches are going to be the most recent, or kind of usually, therefore, the most complex. And that also means they're often usually the most related. So whoever's kind of up here at the final branching, those are the closest related to each other. And a lot of times if you're going from kind of evolution, they're also the more advanced of the group. Okay, so that is essentially how the kind of terms you would need. So those are terms like sister group, but that's not listed in your actual syllabus. So we'll just go with the terms that you definitely need to know. And again, this is probably a little bit of overkill, but where do we get this? So it is constructed based off of DNA similarity. Ideally. So again, you can make it. A lot of cladograms seem to be based more off of morphology, but IB is really hitting that it wants you looking at it at the molecular level. So as DNA evidence, also, once you have the DNA evidence, the other thing you need to think about is this term called parsimony. So parsimony says, here's the evidence, and you need to create the simplest explanation for the seen differences. So parsimony is the simplest explanation for the differences you see. So essentially the one that kind of shows the least speciation events. So you're not going to say, well, these speciated, oh, but then they changed again and became more alike, and then it's like, what's the most logical, the most simple explanation that still explains your finding? And that's kind of the goal, is to do a cladogram that maintains parsimony. Okay, so then this is more about visualizing it. The other thing that you can do with DNA differences, so one, you can build a cladogram. The second thing you can do is you can use it to calculate what's called a molecular clock. Okay, so the molecular clock is essentially looking at the different amounts of DNA change and using that to actually work out a mutation rate or a rate of change. And then you use the rate of change to then estimate the species divergence time. So the first thing you have to do is, from several different species, you need to determine the number of DNA differences. Between different groups of species. Sorry, there's really bad background noise. Sorry about that. They're doing some sort of confusing cleaning on the weekend. Sorry about that. And again, the technique that is often done to do this is hybridization. So if you were to put two single strands of DNA between the species together, how many bases cannot hybridize or join together, and that would tell you the differences. So you're going to have a group, like set of organisms that have some believed shared lineage, and you say, well, these have 6 amino acid differences, these have 9 amino acid differences, these have 15 amino acid differences. So you're going to work that out. Then from that, you're going to calculate basically a rate of DNA change. And this generally helps if you have some sort of dates that you have some idea of when some of the speciation events might have happened. So if you're like, okay, I know these ones kind of branched off at this point and these ones branched here. So I've got some timeline, and then you can kind of use the number of differences. So you don't really need to know how you would calculate that rate, but essentially you have this idea of some sense of time since they were kind of one species, and you have some sense of how many differences there are between them, and then you're going to use that to calculate a rate. And then from that, you can actually estimate divergence times between the species. And again, it's just going to be an estimate. And the reason it's just going to be an estimate is because this rate of change makes the presumption that mutation rate is constant. So it's saying, okay, well, here's the rate, so we can kind of work out that the mutations that accumulated over that time have been at a continuous pace, and therefore that rate works. But that's not necessarily true. So it's not necessarily completely accurate, and that's because mutation rates are subject to a couple of things. So it might be that you have quite a lot of change in a shorter period and then not as much for a while. So it can't fully take that into account. So mutation rates are impacted by 3 things it wants you to identify. So first of all, they're impacted by the population size. So larger populations may have accumulate more, they have more offspring, so there's more opportunities for things to happen in meiosis and so forth. So population size is going to matter. The generation length, so the more reproducing they do, that's why bacteria can become antibiotic resistant in a short period of time, because they have short generations. And then also kind of the extent of pressures, so selective pressures. So if you have really strong selective pressures that very quickly kill off a large number of individuals, it's going to speed up natural selection and therefore kind of make those changes kind of become more common quickly. So those are some factors that kind of speed that up. Okay, so next thing is we're going to look at kind of 2 examples of some things that, given we have cladistics, some things that have happened as a result, so some kind of applications. So these were kind of uses for it, and then these are kind of applications or extensions from now what we now understand of cladistics. All right, so we are on just kind of the last bit of the page. All right. Okay, so 2 things that we're going to look at again, which are just kind of examples. One is the idea of reclassifying taxa. So essentially using this understanding to go back to our fixed taxa and adjusting things to better reflect this. So we kind of said we have a whole shift, and then what do we do with that? And so where we're coming back to is some ways that we've used cladistics to modify fixed taxa. So this is kind of looking at this idea that we haven't necessarily just thrown fixed taxa out altogether. So we do have a whole different way of looking at it, but we can also understand cladistics and improve fixed taxa as a system as well. So one thing we can do is we can use cladistics to reclassify taxa. So we can reorder them into what are called monophyletic groups. So we can say, okay, we're still going to use genus, family, classes, and orders, but we're going to make sure that organisms that we have now put into like a genus are in fact also a clade. So we're going to try to check that that holds up. So we're basically going to reclassify taxa into what are called monophyletic groups, which is basically another way of saying clades. So we're going to try to make sure that when we do use fixed taxa, they also reflect what we understand about clades. So a monophyletic group is one where they all share a common ancestor and all are included. So essentially, definition-wise, it's kind of the same thing as a clade, but we're just using a slide. It's basically saying the groupings need to be clades. So if they're not a clade or some organisms are missing, then we need to reorganize them a little bit. And so essentially, we want none to be separated. So one of the goals is to try to avoid separating members of a clade. So if we have like one family and they are all kind of part of a clade, except these guys are way over here in another family, then they're not grouped correctly. So we want to move these guys that are over here away from their clade into the family that has the rest of their clade. So that's kind of the idea. I don't think you need to know this at any great depth, but the example where this was done was with the figwort family. And sorry, I'm trying to talk over the background, but now I've started. I want to finish this, but they're cleaning just outside the room, so sorry about that. So basically, they looked at their kind of DNA similarity and their phylogeny, and as a result of that, they moved 275 genuses to different groupings. So they moved them out of figworts, some moved into figworts. So they moved a total of 275 genuses, not just species, in order to better reflect their clades. So it resulted in the movement of 275 genera, which is a genus, to reflect their clades. So we still are using the fixed taxa. So it's the figwort family. That's not just a nice term because they're sweet to one another. That's coming back to the kingdom, phylum, class, order, family. So we're still using this term family, but we rearranged some things because they were based on morphology, but it didn't reflect clades that we now understand they belong to. And another term you might see for that movement is called circumspection. And that's just basically movement to reflect phylogeny. So that's just a term for that movement. It's when they're moving to reflect clades. We can refer to that as circumscription. It's just another term for it. So that's one thing that's kind of happened, is that we've still used some fixed taxa, but we've tried to merge the two systems to make that more meaningful. The other thing that is a big change to the fixed taxa was the addition of domains. So that's the other one. So the addition of another taxa. So that is now the largest taxa. So that is now the largest of the taxa. And that came about from kind of 1977, where there's a study. They started beginning in 1977. That's when they discovered archaea. I don't think they fully added it until about the 1990s. But in the 1977, they basically discovered from rRNA a group of prokaryotes that do not share a lot of similarity with general bacteria, and in fact show some characteristics that are more similar to eukarya. So basically, it started in 1977. And at that time, our RNA analysis, which you can imagine is a lot better now, was really just beginning at the time, but just being able to look a little bit at kind of studying that, and we have more evidence of it now. And basically what it showed, that some eukaryotes, background noise is getting to me, some prokaryotes are quite distinct, and they actually have similar metabolism to eukaryotes. Good. So this idea that they actually didn't really fit. So prior to that, we only had kingdoms, and all prokaryotes were in the same kingdom, which was kingdom Monera. So prior to this, it was like everything that's a prokaryote, same kingdom. But then now this is saying, well, wait, some of them are really different. And actually, if you look at it, their metabolism, their transcription, their translation, their replication, their presence of introns. So they have things that actually look like they're more like eukaryotes. And so these prokaryotes became called archaea. And so archaea were discovered in the late 1970s, and within the decade that represented this need for a whole nother domain to be added. So archaea are prokaryotes. They are extremophiles, so they're often found in really extreme environments. They have no peptidoglycan. So those are just a few of the things. I'm not going to go through all their differences, because it doesn't look like in the syllabus you would need to go into specific details about like their ribosomes or their transcription, but I would just note that they do have some metabolic similarities to eukaryotes and also some distinctions from other bacteria, such as not having peptidoglycan. So that's their cell wall, and so their cell walls are unique, and that's kind of a key distinguisher between them and eukaryotes. So as a result, it was added this domain, and there are 3 domains. So kind of the last thing we'll draw today is just this to show the 3 domains. So here's kind of the common ancestry, which would be Luca. And then from Luca, it actually has bacteria as a domain first. Then it actually has archaea and eukarya as more related. So it's this idea of a different pattern of divergence that shows that essentially it did not necessarily devolve into prokaryote, eukaryote, and then go from there. Rather, it branched off into bacteria, and then that remaining group divided into prokaryotes and eukaryotes. So that's a better kind of reflection based on kind of the data. So again, we're still using it as taxa, but it fits. So it's kind of a cladogram-based taxonomic distinction. So then those are your 3 domains. All right, so that one was pretty short, hopefully, because there's not a lot to it. There could just be practice on cladograms, so make sure if you go back over examples that we've done in class and just practice being able to really make sense of them. It's kind of hard to show that in theory, but use the database questions and so forth that are in books to try to make sure you know how to use this, and you should be good. All right, bye.